Start by loading the data. Go to File, Browse. Select the parent folder that contains all images you wish to open. Let's say you have a big study with multiple imaging time points and multiple animal cohorts per time point. If you want to simultaneously visualize all of the animals receiving treatment A, over the course of the study, you'll go to File, Browse, and select the Big Study folder. Living Image will find all of the image files within the parent folder, as well as its subfolders. Select the files you want. If there are several files, you'll select Load as Group. Here, we're opening a single sequence file and clicking Load. In the Tool palette, under Image Adjust, you can zoom and pan. You can also adjust the brightness and contrast of the photograph, as well as the opacity of the luminescence image. You can change the scale bar min and max. Lowering the min allows you to see more of the low-intensity pixels. Changing the scale bar will not affect your quantitation. It only changes the appearance of the optical signal on the screen. You can also change the color scheme. If you want to display the image with a binning other than the one that was used to acquire the image, you can also change that in the Image Adjust tab. You'll notice that the Individual button is unchecked. It is important to uncheck this box if you want to compare multiple images to each other because it thresholds everything to the same min-max scale. If that box is checked, all of the images will be set to different scales and cannot be compared. For analysis, you also have to convert units to radians. Radians is the calibrated unit and is used for analysis, whereas counts are important for determining appropriate signal strength upon image acquisition. Let's draw some regions of interest, or ROIs. A common mistake is drawing the bioluminescence ROI too small. It's possible that due to the default scale setting, you don't see the extent of light scattering through tissue. You want to capture all of the emitted light in your ROI, therefore, you want to draw the ROI big. In this example, we draw 3 ROIs and compare their values. The small oval just covers the BLI signal that's shown on the screen. The large oval covers a more generous area. The box covers the whole mouse. For BLI images, it's okay let some of the ROI go beyond the bounds of the animal. You'll note the 2 larger ROIs have the same value despite being quite different in size. That's because both of them were drawn large enough to capture the entire diffuse signal, and because there is very little background signal in bioluminescent images. The value for the small oval is quite different and doesn't capture the full signal. Let's practice creating templates to speed up the analysis process. If you have a sequence of images open, you'll need to open up a single image by double-clicking on it. Start by drawing a box around the first animal. Remember, the numbers listed here indicate the number of ROIs the software will put on the image. Right-click and duplicate the ROI until you've done so for all animals. Give the template a name and click Save. Prior to drawing these ROIs, I checked the box labeled Apply to Sequence. When I X out of this single image, you'll notice that all of the ROIs I drew in this image have been copied onto all images in the sequence. Depending on the position of the animals from image to image, you may need to manually tweak a few ROIs, but for the most part, they are in the location you need them to be in. If you want to paste this template onto a new batch of images, you will load the images together. Then go to the Tool palette and select ROI Tools. Double-click to open a single image and click the Apply to Sequence button. Choose your template from the drop-down menu and click Load. Before collecting your quantitative data, be sure that your units are in radians. This is the calibrated unit that allows you to compare 2 or more images that were taken with different acquisition settings. Click on the Measure ROIs button. You can either select and copy-paste into a graphing program, or you can save the data as a CSV file which can be opened in most graphing programs.